Hi class, welcome to Module 2A, Chapter 2, Biochemistry Part 4. This is the last part of biochemistry. So we had 4 videos on biochemistry. This one I'm going to talk about proteins and nucleic acids. Proteins and nucleic acids still follow, well, at least in terms of the elements and the atoms that are in proteins and nucleic acid, still follow shown. And with proteins, you include PS, and with nucleic acids, there's a P. Okay, so important, these are organic compounds. Remember what organic means. Or if you are an organic compound, you have a carbon in you, and then you usually have a hydrogen with you. Except, what's the exception to that rule? Do you remember? I can't remember. But anyway, yeah, if you're an organic compound, you're made of carbon and hydrogen. Proteins. Does everybody have a favorite protein? I was thinking soy is pretty delicious. I always tell people if they're trying to put on muscle mass and they want a lot of protein, but they don't want to spend $50 for a big barrel of protein, I say eat tuna fish. Like, it's the cheapest amount of protein. It's got a great amount of protein in it. Like, you don't want to eat it every day, but it's great for protein. So anyway, proteins account for over half of your body's organic matter. Proteins provide for basic construction materials. So you people out there that have worked construction, what's some of the basic construction materials? I don't know. Concrete, rebar, maybe. Anyway, proteins would be analogous to that. So proteins are really important to build body tissues. Proteins play a vital role in cell function. So the cell functions in making things, and oftentimes to make things you need an enzyme. And most of the time, at least for our purposes in Anatomy and Physiology 201, enzymes are made of proteins. Proteins also make up hormones. There's protein hormones and the antibodies. So for example, if you got coronavirus, so you got COVID-19, and your body made antibodies against that virus, those antibodies would be made of protein. And then we talked about this on the first slide. Proteins are shown. Sometimes they're shown S, sulfur. They're built from amino acids. So this is here's a protein molecule on the right, but what is that complex protein molecule built out of? Amino acids. So you guys that have had nutrition, do you have a favorite amino acid? Maybe glycine, leucine, proline. I just saw something fun. This would be a fun reason to have kids. I would name them glycine, lysine, proline, isoleucine. You could name them cool amino acid names. But anyway, a string of amino acids is the primary structure of a protein. And then once that primary structure is built, that sequence of amino acids, due to hydrogen bonds, the amino acid string can form an alpha helix. Let's get right here. Where have you heard helix before? DNA. Exactly. And then once you get these alpha helices formed, you can form other hydrogen bonds between other parts of the amino acids and get this thing called a tertiary structure. Oh, sorry, the alpha helix and beta sheets, that's the other structure for secondary structure. This is the secondary structure of the protein. And then the tertiary or third level of structure. And then the quaternary structure of a protein is just when you have polypeptide, more than one polypeptide joined. So in this cartoon, there's 1, 2, 3, 4 polypeptides, and that would be the protein's quaternary structure. We'll talk more about that later. Okay, so here's, this is a really cool cartoon. I like this because in A, it shows the generalized structure of all amino acids. So this is really important to understand because you'll see proteins again in 202, 205, 218, and beyond. So the basic structure of all amino acids, you have this central carbon, and then there's a hydrogen attached to it. And then, I don't know if you remember your functional groups from a couple videos ago, but on one end of the amino acid you have an amine group, that's NH2, you can see here, and on the other end of the amino acid you have this amino group, or sorry, acid group, carboxylic acid group. And then this R, this thing called R that's attached to the central carbon, that thing can be all different types of groups depending on the amino acid that you are. So let's look at the first one. Here's glycine. You can see it's got the amino end, the carboxyl end, it's got the central carbon, it's got a hydrogen, and its R group is a simple hydrogen. Aspartic acid, it's got the amino end, the acid end, or carboxylic acid end, hydrogen, central carbon, but look at its R group. So it's got the methyl group and the COOH group. And then here's lysine with its R group. Here's cysteine with its R group. So that's the structure of an amino acid. And then here's the symbol. So what's this first one we looked at? Glycine. So the amino acid glycine, it has a 3-letter symbol, GLY, and it has a 1-letter symbol, G. And that's true for all of these amino acids. Okay. And then we said these things. Amino acids have an amine group and an acid group, and then the amino acids vary only by their R group. So each amino acid is completely unique. Protein structure. So we talked a little bit about this earlier. You have 4 different levels of structure: primary, secondary, tertiary, quaternary. And just this simple string of amino acids that has taken its secondary and tertiary structure, that's usually called a polypeptide. And then when you put more than one polypeptide together, that's actually called the protein. If you are a polypeptide, you might have fewer than 50 amino acids. So in this polypeptide, there could be less than 50 amino acids in all this blue coiling here. If you're a large protein, you could have thousands of amino acids. And then the sequence of amino acids will give you a variety of different proteins. So this amino acid, you can see it's got a red, then a yellow, and a purple. Another amino acid could have a purple, then a red, then a yellow. So it's the sequence of amino acids that makes the variety of proteins that you see out. So primary structure, we talked about this. This is the primary structure of a protein. It shows the unique sequence of amino acids, so cysteine, glutamine, leucine, alanine, etc. So we talked about this. There's 4 levels of structure for proteins. There's primary, secondary, tertiary, and quaternary. The secondary structure actually has these 2 substructures, an alpha helix, or you could be a beta pleated sheet, just like in a sheet on your bed when it's pleated. It's the same shape. And here's a picture. So here's the alpha helix, and here's the beta pleated sheet. And the alpha helix and the beta pleated sheet form because of our old friend hydrogen bonds. So remember, hydrogen bonds are between the negative pole of one polar molecule and the positive hydrogen of another polar molecule. So for example, up here, the nitrogen, that's an electron hog. So most of the time that nitrogen is going to hog the electron that it's sharing with hydrogen. So hydrogen will be partially positive. and this oxygen is an electron hog as well, so it will be partially negative most of the time. And then you just have this hydrogen bond between a partial positive charge and a partial negative charge. Okay. So you don't know if you can't really see, you can kind of see the amino acids in here. So here would be a central carbon of an amino acid. And then here'd be the carboxylic end and the amino end and R groups in there. But this cartoon doesn't really show the R groups very well, I don't think. It does show, however, for the secondary structure, that these alpha helices and beta pleated sheets are formed between that positive hydrogen that's linked to the nitrogen on the amino end and the oxygen, the negative oxygen that's linked in the carboxyl or the acid end of the protein or the polypeptide. Okay, yeah. So the secondary structure of alpha helices and beta pleated sheets is by hydrogen bonds, and the hydrogen bonds are between carboxyl and amino groups in the backbone. So so they're just showing the backbone of the polypeptide here. It's just a backbone. It's not really showing the R groups, and it's between a carboxyl group on one amino acid here and an amino group on another amino acid here. Tertiary structure. So now in this cartoon, you can see the beta pleated sheet. So here's the beta pleated sheet. Here's part of an alpha helix. And then those beta pleated sheets and alpha helices take a tertiary structure. So they form another structure, and again, it's using hydrogen bonds. So the tertiary structure is made because you have hydrogen bonds between the R groups of amino acids. So in the secondary structure, it's between a carboxyl group and an amino group of amino acids. In the tertiary structure, you get that structure because you have hydrogen bonds between R groups of amino acids. And then the quaternary structure, the highest level, that's when you get more than one polypeptide subunit. So here's 1, 2, looks like there's one in the back, 3, and then there's one here in the back. So there's 4 polypeptide subunits make a complete protein. And a really well-known example of quaternary structure of a protein is hemoglobin. All right, proteins. Continuing with proteins, there are different categories of proteins. So we're done with the levels, the primary, secondary, tertiary, and quaternary. Now we're going to talk about different types of proteins. There's fibrous proteins. They're also called structural proteins. And so this is the protein on the inside, and it's an alpha helix. And here's an alpha helix in the blue, and another alpha helix in the green. This is a triple helix of collagen. So collagen, you've probably all heard of that. Collagen is the protein, and it forms a helix, an alpha helix, and then you get 3 alpha helices together, and that's collagen. And last I checked, I think there's maybe 20 different types of collagen. So collagen's kind of complicated and cool. So fibrous proteins appear in your body structures. So you probably know that in your tendons you have collagen. In your skin you have collagen. Structural proteins often bind structures together. That's in a tendon, you're binding the muscle to the bone. So binding structures together. structural proteins are usually really stable. And some other examples besides collagen would include keratin. So that's a protein that's in your skin. And maybe you've heard of that. I think they put keratin in skin lotions to jack up the price. All right, the next general category of proteins are globular proteins or functional proteins. That means they usually have a physiological function in the body. So if you are a globular protein, and this kind of looks like a globule, right? Whatever a globule is, but it looks like one. Globular proteins function as antibodies. We talked about those. They can function as hormones, or globular proteins can function as enzymes. And most of the time, if you're a globular protein, you're going to have at least the tertiary structure. Most often, you're going to have the quaternary structure, which is shown in this cartoon of hemoglobin. If you are a globular protein, when you are denatured, that means you lose this form and you can no longer function in your physiological role. So how do you lose your form? A lot of times the environment, like it's too hot or it's too acidic, will cause you as a globular protein to lose your form. There are these pieces on globular proteins called active sites, and active sites on a globular protein will fit or interact chemically with other molecules, for example an enzyme. So here's a table from your book, table 2.6 from the Merriam text. Representative classes of functional proteins, which we're also calling globular proteins. So that would give you the antibodies, we talked about those, hormones, like growth hormone or insulin. You've probably heard of insulin for sure because that's associated with diabetes. That's a protein hormone. And when you take 202, they'll talk about all the different types of hormones, but proteins or hormones can be made of proteins. They can also be made of a lot of other things, like cholesterol, for example. If you're a globular, aka a functional protein, you could be a transport protein. So, for example, hemoglobin transports oxygen. You probably knew that. And if you are a globular or a functional protein, you can be an enzyme, and that's a catalyst. That's something where we talked about this earlier, but an enzyme will bind a substrate, or you could say it's a reactant, and will trick that substrate or reactant out and make a product. And then it's not here, it's not in the table, but there's these other cool proteins called stress proteins. And stress proteins are called chaperones. So for example, if they're studied, when I took biochemistry, one of our professors studied stress proteins in plants. And so when you heat shock or you heat up a plant out of the range of temperature that it likes, it starts making these stress proteins, and stress proteins are commonly called chaperones. Here's the enzyme picture that I wanted to show you guys. Enzymes, for our purposes, almost always are made out of proteins. So this is just a big purple blob. It doesn't show you its primary, secondary, tertiary, or quaternary structure, but it's in there. So this is an enzyme, and you have these areas here called an active site, and the active site is where you fit the substrates. I think in chemistry they call these things the reactants, but in physiology more often we call these things that fit into the active site substrates. So in this cartoon it shows 2 amino acids, so you're trying to build a polypeptide, so you want to link your first 2 amino acids together. So the amino acids will fit in the active site of this enzyme, and then once the amino acids are in the active site, you call that the enzyme substrate complex. And then what happens is the enzyme substrate complex undergoes kind of a shift, so it moves around a little bit, and you form a bond. And there's a whole bunch of other biochemistry, but you form a bond here, this green thing, between the 2 amino acids. The energy is absorbed, so energy goes in here to form this bond. and water is released. So hopefully if you were paying attention to the previous lectures, you'd know that when you synthesize or you bring two substrates together and water is released, that's a dehydration synthesis. That's the type of reaction that it is. And then you release your product, and you have a peptide bond. That's peptide bond is the bond between two amino acids. Biological catalysts are usually proteins. They can be RNA, but usually they're proteins. Enzymes will increase the rate of chemical reaction. So if there was no enzyme here, these two amino acids might get together, but it might take 1,000 years, where it takes 1,000 nanoseconds if there's an enzyme around. Enzymes will bind to substrates at specific active sites. So if these two brown things did not fit into the active site of that enzyme, then you wouldn't be able to form a bond between those. So there's specificity involved in enzymes. Enzymes, they can do this over and over again. They can make thousands of dipeptide bonds. So you can use an enzyme many times. You don't just use it once and then it's done. You can recognize enzymes most of the time by their suffix ase. So for example, hydrolase, oxidase, etc. Okay, that was it for proteins. Now let's look at nucleic acids, DNA and RNA. So here's a cartoon. Here's DNA, deoxyribonucleic acid. Here's RNA, ribonucleic acid. And then the bases that form DNA here, so here's thymine. There's its chemical structure. Here's its name. So this would be a thymine here in the DNA double helix. You might remember this. T always binds with A. C always binds with G. So here's the chemical structure for adenine. And you can't see it, but in all these nucleic acids, there's chone in there. Because at each one of these little corners, I don't know if you remember, I told you guys this before, at each one of these little corners, there's a carbon. And where there's a carbon, there could also be a hydrogen there. So carbon, hydrogen, oxygen, and nitrogen chone. So here in the DNA double helix, here's adenine, because T and A always form hydrogen bonds with each other to help create the double helix. And then cytosine in blue and guanine in this fluorescent green, they always form hydrogen bonds. And when all these bases, A, T, C, and G in DNA, when they form hydrogen bonds properly, they make the DNA double helix, which you can see in the cartoon. Here's RNA. So RNA is not a double helix. It's single strand. And instead of, well, RNA and DNA both have cytosine. RNA and DNA both have adenine. RNA and DNA both have guanine. DNA has thymine. RNA does not have thymine, but it has something different that's kind of close, uracil. So nucleic acids in general, DNA and RNA, are the largest of the 4 major biomolecules. So what are the other 3 biomolecules that we just studied? Carbohydrates, fats, proteins. Nucleic acid make the 4 major biomolecules. DNA makes up genes, and DNA and RNA are made of chone P. So chone with the P, which stands for phosphorus. And it doesn't show it in this cartoon, but this gray ribbon, this is the backbone of DNA, and it has phosphate in it. Okay, nucleic acids are, well, nucleic acids include DNA and RNA. If you're DNA, you are a double-stranded molecule containing A, always binds with T, G always binds with C. If you are RNA, you are single-stranded, and instead of T, you have U for uracil. If you are are RNA or DNA, you have a nitrogenous base. That's all these guys. A, G, T, C, and U. They're all nitrogenous bases. And they are linked to this thing called a pentose. That's a 5-carbon sugar. And that's linked to a phosphate group. And I think I have a, you can't see it in this picture, but I have a picture. Next picture. Yeah. So in this cartoon, here's the base. So this is adenine. So if it's adenine, this could be DNA or RNA, because DNA and RNA both have adenine. So you have the base, adenine. You have the pentose sugar. So pentose is 5. 1, 2, 3, 4, 5 corners. So this is the pentose sugar. And then here's the phosphate group. So this is an adenine nucleotide. You could have a guanine nucleotide. You just put guanine here if you had a guanine nucleotide. And then the phosphate and sugar would look the same. Okay. So here's another cartoon of DNA. We just talked about this. This actually shows the hydrogen bond. Remember, hydrogen bonds can be shown like this, like little dashed lines, or they can just be shown as dots, like it is in this cartoon. So this shows a hydrogen bond, A and T, C and G, and you get the double helix. And then it doesn't show it, but this ribbon contains the deoxyribose sugar and the phosphate group. So DNA is genetic material in the nucleus. You knew that. The DNA provides instructions to make every single protein in your body. We already said it forms a double helix. And if you are a single mother cell and you want to make 2 daughter cells with the exact copy of DNA, then you have to replicate your DNA. And then you give 1 replicate of DNA to one daughter cell, and you give another replicate of DNA to the other daughter cell. Okay, so this is RNA. We've gone over this. It still has that sugar phosphate backbone, this gray thing that they didn't draw out. And then it's got its 4 bases: cytosine, guanine, adenine, and uracil. Uracil instead of thymine. Remember, DNA has thymine, RNA has uracil. So RNA carries out DNA's instructions for protein synthesis. So we'll talk about this later, but the central dogma of biology or genetics is DNA gets transcribed into RNA, and then RNA gets translated into a protein. That's the central dogma. So RNA carries out DNA's instructions for protein synthesis. RNA is created from a DNA template, and we'll look at that in more detail. RNA is organized by complementary bases to form a single-stranded helix. So it says there's complementary bases, but the bases, so you don't have 2 strands of RNA together with C binding with G, for example. What you have, complementary base-wise, is if on your DNA that you're transcribing, if you have a G here in your DNA, that will get translated to a C in RNA, and then that makes that a complement. G and C is a complement. Okay. There's 3 varieties of RNA. There's messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, rRNA. And there's DNA. Okay, the nucleic acid, ATP, kind of a big deal, kind of important. It's the energy currency of the cell. So check it out. In A, you have ATP, adenosine triphosphate, coming in. ATP provides the energy needed to drive energy-absorbing chemical reactions. So if you want to hook A and B up, you need to put energy into that reaction to get A and B to hook up and form AB. And that energy you put in is ATP. And in this cartoon, you just used one phosphate from ATP. So adenosine triphosphate now becomes adenosine diphosphate. So that's chemical work. ATP does chemical work. ATP does transport work. So here's the phospholipid bilayer in your cells. So here's inside the cell. So the intracellular fluid inside the cell. And out here is the extracellular fluid, ECF. And then you have this membrane protein, and you're trying to get this blue ball from the intracellular fluid to the extracellular fluid. And because you're moving this blue ball against its concentration gradient, so you're going from low concentration of the blue ball to high concentration of the blue ball, that's against the gradient, you need ATP. And this membrane transporter will use ATP and move that blue ball from the inside of the cell to the outside of the cell. And you will release adenosine diphosphate. ATP can do mechanical work. So check it out. Here's a relaxed smooth muscle cell. You find smooth muscle cells everywhere in your gut, especially. It helps move things along in your gut. So the smooth muscle cell is relaxed. But let's say you have some food in your esophagus and you need to contract those smooth muscle cells to get the food to go down your esophagus. That's mechanical work. ATP can do that for you. ATP can cause a relaxed smooth muscle cell to contract, and if it contracts in sequence in your esophagus, you just swallowed your food. Sweet. And we talked about this. ADP accumulates as ATP is used for energy. So you can see here you're using ATP in chemical work, you're using ATP in transport work, you're using ATP in mechanical work. And as you do that, this molecule ADP is accumulating. But your cell has ways to put another phosphate on ADP and make more ATP. Here's adenosine triphosphate. It's all its glory. So here's the base. You can see what it's made of. I like this cartoon because it actually shows the carbons here in the corners. And it shows the hydrogens too. So here's carbon. Remember, carbon can take 4 bonds, 4 single bonds, or 2 single bonds and 1 double bond, but a total of 4 bonds. So here's your carbon. It's got a bond here. It's got a double bond here. 1, 2, 3, 4. So this is the base. Adenosine is the base. Here's the sugar, ribose, and you can see it's attached to the base here. And then the sugar ribose is attached to these 3 phosphates. And these phosphates have super high energy bonds in them. So when these bonds, they break, a lot of energy is released, and then you can do a whole bunch of work in your cell. And here is a cartoon showing the hydrolysis. So remember hydrolysis reactions, you need a molecule of water to break a bond. So this molecule of water went in to break that terminal phosphate off your ATP.